So welcome to the class of the Introduction to Drug Designing and Drug Discovery. I'm Dr. Sukhwant Singh, professor at Sagar Institute of Research and Technology, Pharmacy, Sage University, Bhopal. In today's lecture, we will have a brief intro and a brief discussion on the process of drug discovery and drug designing. The objective of this particular lecture is to give you a brief orientation about uh, how about and what all is to be done when you try to start your dissertation or you go into the process of drug designing and finding out something new. And now before we do that, I would like to have a brief discussion, or let us say a small glimpse into the process of drug designing we'll try to have, and we will discuss with your participation, we'll come to understanding regarding this particular topic. So when I begin, I usually start with a story. That is how I usually conduct my classes. But since we all are, already postgraduates and doing our PhDs, I would like to start this presentation with a small quiz or a small questionnaire. I think the screen might have changed now, and three personalities might be visible on your screen. Are they visible, please?
Yes, sir.
Yeah.
Yes, sir.
So the question is this, that can you recognize these ladies and gentlemen? Anyone?
Sir, Dr. Barje.
And more. Please keep on guessing, because I'm sure most of us will not be able to understand and recognize them. It is actually not Professor Dr. Alfred Berger. I'll cut to the chase. They are Professor William Campbell, Satoshi Omura, and Yo-Yo too. And a great thing about these ladies and gentlemen is that we can see from the photos that they are almost octogenarians, or let us say at least septuagenarians. They have all crossed their ages from 75 through 80 years. Most of them are in the same age group when these photographs were taken. And what did they achieve? That is the main question here. So again, I'll tell that these people actually won the 2015 Nobel Prize in Medicine. So Professor Satoshi Omura, Yo-Yo too, and Professor William C. Campbell, they have won the 2015 Nobel Prize in Medicine. 2015. So that is around 8 or 9 years ago. And what did they discover? They discovered ivermectin and artemisinin. So, and these ivermectin and artemisinin are these complex structures that you can, which are visible on your screen right now. And these scientists were already grandparents when they went into the process of discovering these items, these particular drugs, ivermectin and artemisinin. So they were involved in the research, and they were having the bent of research, and they wanted to find something good for the humanity. These ivermectin and artemisinin are actually the drugs which are anti-malarials in nature, and these drugs have got their profound activity on the falciparum genus of the Plasmodium. So these both drugs, ivermectin as well as artemisinin, are from the nature's origin, and these professors have invested a lot of a big part of their life in discovery of this particular agent. What I mean to say is that since most of us have started off our PhDs, some of us have started a bit later than that was expected. But I want to just, through these slides, I want to convey that there is no age for discovery. There is no age for working. There is no age for working hard. Now, with that note, I welcome you all once again to the PhD program, and I request you all that if these people can achieve this feat at an age of above 80 years, let us just imagine that what wonders we young minds can create. So with that note, let us start discussing about the drugs, that what are the drugs. So by the definition, drugs are a medicine or other substances which has a physiological effect when ingested or otherwise introduced into the body. This is the general definition. that we usually come across. And the objectives of tracking drugs are either treatment of disease or relief from symptoms. Sometimes they also provide supplementary nutrition, etc. There are so many other objectives when individual ingests certain drugs. Now, from the introductory slides, when I talked that this ivermectin and artemisinin were from the nature origin, so we all know that the drugs usually come from either plants or from the animals or the minerals. Some also have their sources from the marine sources, and most of the drugs that are coming into the market these days, they are either synthetic or they are also sometimes derivatives of natural drugs or semi-synthetic drugs that are coming to the market these days. So we have got a huge plethora and a huge basket, or let us say a big range of origins from where we can obtain drugs. For example, if you look at the next slide, you can see that various drugs are obtained from various other sources from the nature. For example, if you look at examples from the plants, we can see that the leaf, bark, fruit, and seed of the plants, respectively digitalis, cinchona, and opium and iserin, are used for getting various drugs like digoxin, quinine, morphine, and anticholinesterases, which are used in variety of diseases. Like, for example, digoxin is used in CHF, quinine is used in malaria, morphine is a very good known analgesic, and anticholinesterases are used in the diseases like myasthenia gravis. So if you look at the just one source, that is the plants, we see that all the parts of the plants are used for various kinds of drugs. So this kind of mining usually exists, and we can get so many active principles from various parts of the plants also. For example, there are certain drugs or certain sources where a single plant is able to produce different kind of molecules from its various sources. For example, if you take neem's leaves have different components, neem's bark has certain other components, neem's root has certain other components, and these are used for treating various other types of diseases. So if you look at the nature, we get a lot of resources from where we can look into getting a new drug. For example, you look at this Nicotiana tabacum, Atropa belladonna, or Papaver somniferum, wherein we use different kinds of extracts from various drugs. For example, you use exudate from the Papaver somniferum, right? And hence so on, we can have different kind of sources from where we can get drugs. We also have certain examples from animals, like hormones can be obtained from animals like insulin, oxytocin, epinephrine, and enzymes are used from animals like pancreatin or chymotrypsin, etc. And certain animal extracts like the cod liver oil and heparin, etc., are used for getting drugs. These are the pictures of some examples of the drugs that have been obtained from the animals. You can see at the. Excuse me, please. Please switch off your mics. I request you all.
अभी चेंज कर
Yeah. So there are certain important pharmacological active principles that are found in the plants. Like from plants, you can get alkaloids. From plants, you can get glycosides, oils, resins, gums, and tannins, which are exploited and used for finding out various active pharmaceutical ingredients, or various resources or leads can be found out from these plants. Now, having said that, a question that comes into the mind of a researcher, that how to proceed, because the iterative process of drug designing is a big step, especially when you consider with respect to your PhD thesis. For example, the students from pharmaceutical chemistry, they will definitely look into this aspect, that having knowledge of all these fields, it's very difficult for us to predict or take up one line wherein we can go in for drug designing. Definitely, if we have some sort of prior information or prior art is done in that area, we can definitely proceed. So let us now just discuss the iterative process of drug designing. If you look at the historical purview, most of the drugs that are available today in the pharmaceutical market have either been discovered by the active ingredients that were available from the traditional remedies or from the accidental discovery, that is serendipity, which is accidental discovery. And the antibiotics like penicillin, streptomycin, cisplatin, vinblast in the anti-cancerous drugs, these were all found out accidentally. If you look at various type of inspirations, we can always find inspiration from our homes and our kitchen. Like we can get home remedies and drugs from our kitchen, like turmeric is there and ginger is there. For example, turmeric is used as an antiseptic, which is given with milk for body pain, and it is also a very good anti-cancer agent. Similarly, ginger. We can, as a scientist, also look into the various parallel spaces that are available to us. For example, we can look into various sources from Ayurveda. We can look into the drugs that are available in Unani. We can also look at Siddha. Of course, there is homeopathy. Then the additional areas where we can look into are Chinese system of medicine, the Oriental system. We can also look into the drugs like drugs from naturopathy and other similar sources or sources of similar nature can be explored when we want to look into some sort of prior information or prior art, or also some sort of inspiration. Now, the modern drug discovery process is an iterative process, which is usually what we have heard from our graduation and post-graduation years, that usually a drug takes 14 years time to come from the desk of the chemist till the desk of from the discovery till the marketed preparation. Usually a drug takes around 14 years. That process is, of course, it is true, but with the advent of science and development of the science knowledge and various other computational facilities, this process is now coming down to around 7 to 8 years if somebody works very hard and all the positive green signals are obtained. Usually 7 to 8 years time, a drug can reach from the desktop of a laboratory till the chemist's shop. Now, there are three aspects to the modern drug discovery process. These are lead drug discovery and research, then iterative process, and preclinical R&D before you come into the investigational new drug filing. So the lead discovery process may involve biological discovery, high volume screening, combinatorial chemistry, rational drug designing, which includes structure design as well as informatics. After we have crossed these lead discovery process, we come to a lead series where we get a lead, and from the lead we can either synthesize or identify the main molecule on which the discovery has to be carried forward. And after we get various derivatives of the molecule, we can go for the preclinical R&Ds, provided that we are getting positive results. And in the preclinical R&D, we usually do the toxicity and efficacy studies. And upon getting a sufficient and a positive response and results from the preclinical R&D, the drug is further proceeded for the investigational new drug or the IND category. Now, this process will take around millions of rupees, I guess. If somebody is from the industry, they'll be in a better position to give the exact statistics, but it takes a lot of money as well as a lot of hard work on the base on the part of the scientist who's working in that area. Now, the word that we need to now look is lead. So by definition, what is a lead? Any species or a chemical structure or a ligand that is able to give a viable desired biological activity on a defined target can be termed as a lead. Now, I have taken up 3 words here, and the 4th one will be another important word. I'm going to underline this. First word is viable. Second word is desired. And the third one is biological activity. So here I am actually, ruling out the word serendipity here, because when you start a process of drug designing and you have fixed up a target, because, we have defined a target here. So when you define a target, for example, you have identified a particular gene or a particular enzyme or a particular cycle. So that means that you now have you have defined a target against which you need to develop a molecule or a drug for getting a desired action. And therefore, after fixing up this defined target, when you get a viable and a desired biological activity, then that chemical structure or that species or a ligand will be termed as the lead molecule or a lead, in the simple words. Now, the next step is from going from lead to hit. Now, many of my students, when we discuss in the class, they ask, "Sir, word lead, why have we used this word lead only?" So this lead, usually, if you have seen some crime reporting or some, serial of that nature, तो उसमें जब इंस्पेक्टर फंस जाता है ना, तो बोलता है, "सर, मुझे लीड नहीं मिल रही है इस केस की." तो वो लीड का मतलब, या that lead here means exactly way forward. When you want to proceed in certain direction, you don't get any way forward, then we say that we don't get a lead. So here, this lead directly means a way forward into the iterative process. Now, what is hit? So any derivative of a lead or the lead itself, when is able to give a predefined and desired biological effect on the receptor, then it is termed as a hit. So of course, we have to have a predefined effect as well as a desired biological effect on the receptor against which the action or the activity is to be carried out. So if we get a desired activity and a desired biological response, then we call that lead or its derivative as a hit. It gives us a hint that the scientific study can probably reach a successful destination. Because, just as I said, that a lot of money as well as a lot of hard work is to be expected from a scientist at this particular juncture. Therefore, getting a hit is very, very essential before we can, go all ahead. Now, establishing the biological activity, we can, have various models. Now, since the science has developed so far, with that we can go for variety of activities and variety of methods from where we can establish the biological activity. For example, we can go for in vivo studies wherein the experimentation is carried out either using a whole living organism or a part of the organism, which gives us information about the metabolical profile of the drug, the toxicological property of the drugs, as well as the drug interactions. We can also establish the biological activity through ex vivo studies, where the experimentation is either carried out on the tissue in an artificial environment outside the organism with the minimum alteration of the natural conditions. This can also be carried out, and we can also have these days in silico studies, wherein we can always look at certain models and certain simulation studies in which we can predict through various systems the activity that can be obtained from a particular system. Then we come to clinical trials. The clinical trials are nothing but set of procedures in medical research and drug development, where we study the safety and efficacy of new drugs. This step is very, very essential to get marketing approval from regulatory authorities, and usually it may require up to 7 years' time. Now, there are various phases to clinical trials, as you all are aware. First one is the phase 0 studies. This is the recent designation. This is also known as the human microdosing studies. This is concerned with the preliminary data on the drugs, pharmacodynamic as well as pharmacokinetic study profiles. And this also is concerned with the efficacy of preclinical studies. Then the next phase is phase 1. Phase 1 is concerned with the human toxicity. And we also study about the dose ranges which can be tolerated by the particular organisms. And we also look at the pharmacology as well as dynamics of the molecule. So this is phase 1. In phase 2, we are more concerned with the safety, efficacy, drug toxicity, and drug interaction. In phase 3, we are concerned with the safety, efficacy, then comparison with other drugs, and lastly, the package insert, because this is a step after phase 3 when the drug is going to be launched into the market. And lastly, that is phase 4, which is also termed as the post-marketing surveillance or the PMS. This is usually designed to detect any rare or long-term adverse effects and adverse drug reaction monitoring as well as pharmacovigilance. So phase 4 is usually carried out after the drug has been launched into the market to look at the long-term effects of the drugs after coming into the market and its various effects on the race. So now if you look at the whole process, the drug discovery and development timeline, if you look at it, when we talk that usually 14 years time is taken for a drug to come from the desktop of the discoverer till the chemist desktop. This is the whole process. In the first process, before the preclinical studies, usually a scientist starts with 10,000 compounds, which this is the data which I have taken from the US FDA site. So they predict that when the process of drug discovery starts, usually 10,000 compounds start the race, and around 250 compounds complete the preclinical trials, preclinical phases. And this process usually takes around 6.5 years time. Out of the 250 compounds that have completed the preclinical studies, only 5 compounds are accepted into the clinical trials. Remember, this is the phase where the various types of clinical trials of 0, 1, 2, 3 trials will be completed. And this process usually takes around 7 years of time. And after the completion of this 7 plus 6.5 years, that is around 13.5 years, FDA reviews after you submit the dossier and all the study data to the FDA. FDA reviews and takes around 1.5 years to review these 5 compounds that have gone to the table of FDA for review. And ultimately, after the complete investment of this 14 years time, only one drug gets the approval from FDA. So from 10,000 to 1, an investment of millions of dollars and 14 years, around 14 years of time, only one drug out of 10,000 compound reaches or sees the end of the tunnel and the day of light. So this is the complete drug discovery and development timeline that we come across. And with that, I thank you all for attending this particular class of brief introduction to the drug discovery. I'll just stop the recording here so that now we can discuss various aspects, and we can talk about various queries that if you have any.